The group in Lisbon further studied what happens if you knock out HRK1, that strain, as you can see here, it grows very poorly in the presence of even low amounts of acetic acid. And you compare that to a wild-type strain. Then they studied, comparing those two strains, how the phosphoproteome, remember HRK1 is a protein kinase. So by studying the phosphoproteome, you can see what the differences are in an HRK1 strain and in a wild-type strain for the phosphorylation of different proteins and how that actually worked. And they confirmed that of the proteins that have a different level of phosphorylation upon exposure to acetic acid, that 20% of those are actually linked to the HRK1. So this really shows the important role of HRK1 in the regulation of acetic acid tolerance, coming back to phosphate metabolism, translation, cellular transport, and the general stress response. Despite the fact that we really learned a lot about acetic acid tolerant in yeast and that we have improved strains, we still wanted an additional level of acetic acid tolerance. We wanted something more. And we looked towards evolution as a way to get cells that are more acetic acid tolerant. And one way to do that is simply by doing a sequential batch culture. Take your yeast, put it in a batch. At the end of the exponential phase, you pump out your medium or you transfer to your next shake flask. And again, you add fresh medium and you continue this cycle. If you do that in the presence of acetic acid, then indeed you get strains that grow faster at high acetic acid concentrations because that's what the selective pressure is on. We did this. And here you see that we increased the acetic acid concentration at actually a low operating pH all the way to 6 grams per liter. And in the end, the yeast were able to grow. The wild yeast would not have been able to grow under the same conditions. So it worked, but this phenotype is not constitutive, but hyper-inducible. What does that mean? Well, I'll show you here with an example just of the wild yeast. That if you take a yeast that has been pre-exposed to acetic acid just once in one flask, and you transfer that one to a new flask, and you analyze growth, you actually get this growth curve. And if you take a yeast that has not seen just before acetic acid, and you transfer that to a flask with acetic acid, you get that very long latency phase. The evolved strains still had this, but were tolerant to concentrations of acetic acid of a much higher concentration. So they were more tolerant, but they still needed this induction. Again, for industrial application, it's not really handy if you first have to expose your cells to acetic acid a bit, let them get used to it, and then put them in a large fermenter. You would want them to be tolerant from the start. So another approach is to actually do that evolution in an alternating on-off setting. First, a flask without acetic acid, then expose them to acetic acid of a certain concentration. Then the cells that can grow there actually have a competitive benefit. But then we all switch them off again, basically, by not giving them acetic acid. And go to a next flask. Every now and then still increase the acetic acid concentration. We have some ambition. And keep this up for half a year, and see what comes out. We did this, and this is just the reference strain, so the original strain, and this is the evolved mutant of that, following that protocol. We screened them for acetic acid tolerance in microtiter plates with very high amounts of replicates, and then actually you get the outcome as seen here. On the x-axis, the acetic acid concentration, at the y-axis, the optical density at the end of 3 days. And you see that where the reference strain at concentrations of acetic acid around 0.9% completely stops growing, that the strains that were evolved in this way can, when transferred from non-stress conditions to acetic acid, because that's what we do here, can actually grow until dramatically higher concentrations of acetic acid. And we've also shown that it's actually an increased percentage of the cells that starts growing here. And a very highly increased percentage of the cells. But then we want to know, actually, because the aim of the study is to also understand what is going on. So of 6 parallel evolution lines, in our experience in Delft, we really see that when you sequence multiple parallel lines, you have a much higher chance of understanding what is going on and by finding the relevant hits. We sequenced those, but we still got 10 to 30 mutations per strain. So we needed to narrow it down further, and for that we used crossing and sporulation. And in the first, you can both simultaneously find out how many mutations are responsible for the tolerant. Is the mutation dominant or recessive? Turned out to be recessive in this case. And when you have, after multiple rounds of crossing and sporulation, still cells that are acetic acid tolerant, you can again sequence those. So we did that by making multiple segregants, screen every time for the tolerant ones, similar as we did for the tolerant wild strains, but this is with evolved strains, so you have less genomic differences than a completely different wild strain. We tested them, and we actually found some of the segregants that have exactly the same phenotype as the originally evolved mutant. Those we have resequenced. There's only a very few number of priority targets left. The rest has been diluted out by the crossing and sporulation, and we're currently reverse engineering those targets. Like other speakers, for IP reasons, I'm not mentioning the specific genes yet. To wrap up, I want to introduce one other way to deal with acetic acid, because you can make your strain more tolerant to it, but what if you could use it as a carbon and energy source and actually make product out of it? We have thought of converting acetic acid actually to ethanol and make a problem into something nice. If you have a bad wine, it goes a different direction, so it already indicates this must be a challenge. It probably doesn't come for free. But if you would put some electrons in acetic acid, in theory it's possible. In yeast, acetic acid can be activated to acetyl coenzyme A, and then there are genes in other organisms, not in yeast itself, that can actually take the acetyl coenzyme A and convert it to acetaldehyde, putting in some electrons, and then again putting some electrons from acetaldehyde to ethanol. So there is a pathway possible as long as we express this in yeast to get acetic acid all the way to ethanol. Yeast normally turns glucose into glycerol to get rid of some excess NADH that actually comes from the formation of biomass. When yeast grows, it has excess NADH. That goes to the formation of glycerol. And at industrial scale, roughly 4% of the carbon goes to glycerol formation. So 4% doesn't sound like a lot, but we make roughly 100 billion liters of ethanol per year. 4% of 100 billion liters times 50 cents per liter is in the order of 1 to 2 billion euros, so that's definitely relevant. So we could, if this works, increase the ethanol yield by 6% by replacing this by that. We tested in a batch. This is the wild type condition where the cells grow, consuming the glucose. They actually don't do anything with the acetic acid in that case, and they make ethanol. They make ethanol, they make biomass, and they make their glycerol. When we knock out the formation of glycerol, the cells cannot grow anymore normally because they can't get rid of their excess electrons. But when we provide the cells with the gene from E. coli and acetic acid, you actually see that the cells grow, consume the glucose, no longer make glycerol, and they still make ethanol. And actually, under laboratory conditions, which is more optimized for growth and therefore make more glycerol than in industrial conditions, you make 13% more ethanol under those conditions. We currently, together with a company, transferring this knowledge to industry. If we look at the original plans in the work packages, then you see, as I actually told, there's a few of the work packages that are still ongoing. The project, because of a late start of some of the people, is still ongoing. I have good hopes that for at least a few of those we can really determine the underlying molecular basis. And then actually to apply all of those together, that might take a little bit more time, but I'm convinced that with the partners and how we are working together that we can also do that after the end of the Intact project, if needed. With that, I would like to conclude that we have an improved understanding of how acetic acid affects processes actually, the impact on the single cells, genomics from the transcriptomic and the proteomic work, the fact that you can induce acetic acid tolerance. I don't know if it's interesting for industry directly, but it's very important to know. We directly have strains with improved tolerance to acetic acid, wild variants, And with the dramatic improvement in synthetic biology tools over the past few years, it really has become possible to transfer things you find out in a laboratory strain quite easily to other yeast strains. So they themselves might become interesting platforms as well. Evolutionary engineering can dramatically improve constitutive tolerance to acetic acid, and the reverse metabolic engineering of this work is still ongoing. With that, I would like to thank you for your attention, and of course, I'm available for any questions you might have.
Okay, do we have any questions? Yep.
One question to ethanol red. It was a dry yeast, I think, you used.
This is when it's inoculated in there. It's no longer the dry yeast, but it's often sold as a dried yeast. But here it was.
Yeah.
They were all pre-cultured under identical conditions, and an identical amount of cells was transferred to that screening.
You told about the diverse ratio between living cells and cells they do not grow.
It's actually, they're all alive, surprisingly, but it's the ratio between growing cells and non-growing cells. They don't die.
Under these stress conditions?
Yes.
Okay.
So and the ones that don't grow don't die immediately. They only die after days later.
And without these stress conditions, they are better growing. More cells are growing.
So they all grew more or less the same. That was the gray bars. In the absence of acetic acid, and in the presence of acetic acid, we saw the huge differences.
Okay, thanks.
Hopefully very soon a publication on that will come out in FEMS yeast research.
I think the gentleman at the back was first, sorry. Try and do it in order.
I have a question regarding the induction of acetic acid tolerance. So it takes like 40 hours. So are you sure that this is a typical induction? So
no. So the difference, the induction actually happened in the flask before that. What you saw there was that the induced cells immediately start growing. The non-induced cells show that latency phase of 40 hours, and then only when they start growing, it's actually only a very small fraction of the cells that started growing from the beginning there. So that's definitely not classical induction.
Okay, yeah. So, for example, for solvent tolerance, it was also seen that other mechanisms are involved, like mobile DNA elements jumping around in regulatory genes, or
This is not mutations or not something like that, because we've taken cells actually that were induced, also in those, put them under non-stress conditions, and the next one after that you see exactly the same. So you're not enriching for mutants or for variants with different plasmids or something like that.
So there are no genetic differences between the growing and non-growing cells.
Okay, one at the front.
It's actually a very similar question. So it means that this ethanol red strain has a natural diversity or a mechanism to increase diversity which the others do not have?
There is something which is, I think, at a non-genetic, so epigenetic, we don't know exactly what yet, I would love to know, but I don't know, level that determines which cells start growing or not. I would not say that ethanol red has a diversity. I would say that the fraction of cells that have something that helps with the tolerance, with the start growing, is higher in actually the other strains. So I would say ethanol red has less of whatever makes them tolerant to acetic acid.
But you see that in your ethanol red strain, you have some which do not grow at all,
but I think
some which start late.
But that's simple stochasticity, because if only 1 to 10 out of 10 million cells or 1 million cells start growing, then the chance that when you inoculate a flask, some have none of those and some have 2. So that's really stochasticity at the low numbers.
Okay.
Okay, any other further questions? No, that's great. Thank you. Okay, so the next project is ProAkim. Let's find this one. Aha, you're going to go live, are you? Yes. Great. Okay. asset. That one?
This one's PPT and this PPTX. And
oh, okay. And do you want to run it from there?
yeah, sure.
It's up to you if I'm not sure whether. Okay.
Okay. Yes. Good day, everybody. I'm happy to present our results of our project ProAkim here. In fact, when I was preparing the presentation, I was tempted to subtitle the presentation by how discouraging science can be, or the pursuit of unhappiness. Well, I didn't so after all, because at the end of the day, in fact, really at the end of the day, we finally succeeded in reaching what we have aimed at, but in fact, it's a story of 3 years of almost discouraging research and drawbacks and failures, but you have never to give up, and this is what we did. So the aim of the project was to produce these amino alcohols. These are important chiral entities in a series of pharmaceuticals. They serve as chiral building blocks in the synthesis of non-natural amino acids, and also in the production of oxazolidinones, which themselves are either chiral syntons for pharmaceutical production, or they serve also as chiral auxiliaries. The idea was to establish a library of different versatile amino alcohols by means of introducing threonine aldolase coupling as a new synthetic means of producing these compounds. And what makes this approach so interesting is that we can vary the aldehyde component, we can vary the amino acid, and what was so challenging about this project was that there has been some previous research done on varying the composition of the aldehyde, but not yet of the amino acid. The only amino acid previously being researched was glycine, and our idea was to use also others like alanine or phenylalanine and others, and produce from these entities oxazolidinones or these chiral amino alcohols. This aim was to be achieved by identifying new threonine aldolases supported by direct evolution and site-directed mutagenesis, and also with the help of in silico enzyme design. The enzymes were to be characterized and then transferred to process engineering. That means to provide sufficient enzyme for laboratory and larger scale experiments in the gram to kilogram scale, and also to produce the amino alcohols. What is necessary in this context is to develop a method to shift the reaction equilibrium to the product side, because the enzyme actually catalyzes the cleavage of these entities to the substrates, and therefore we needed a method to remove in situ the product from the reaction mixture. And after all, we were interested in the possibilities of a process intensification through the use of microstructured reactors as a novel principle in biocatalysis. We are starting from threonine aldolase from Thermotoga maritima as a model enzyme. This enzyme has the advantage of being highly stable, and it can be obtained from recombinant expression from E. coli in good yields, and also the three-dimensional structure is at hand. And from this 3D structure, 13 promising amino acids have been identified and subjected to mutagenesis, and a small library with 96 single mutants has been established. These were compared with the properties of the wild-type enzyme. This has been done using a photometric assay, using threonine cleavage. This is the usual test for this class of enzymes. And to cut a long story short, none of the mutants did or was superior to the wild-type enzyme, neither with phenylserine nor with metaraminol. The metaraminol is typically not cleaved by the wild type enzyme, and the question was whether it possibly is cleaved by the mutants. The idea was to extend a substrate range, but here no further